Hello, and welcome to this video on making glucose in C4 and CAM plants. At the end of this video, you'll be able to explain why CAM and C4 adaptations are necessary and describe how CAM and C4 plants separate oxygen from rubisco during the production of glucose. So rubisco is a necessary protein involved in the Calvin cycle, but it has a side reaction that plants have to deal with in order to maintain the efficiency of that enzyme. So we know that rubisco is the enzyme that takes carbon from the atmosphere and combines it with ribulose 1,5-bisphosphate in order to generate the 3 carbon molecules necessary for the formation of sugars in the Calvin cycle. The problem is rubisco is an ancient enzyme that evolved when there was far less oxygen in the environment, when we were really in a kind of methane world full of carbon dioxide, but not a lot of oxygen. So structurally, rubisco can accept carbon dioxide as a substrate, which is great because it can use that to generate the carbons for sugars. But it can also take oxygen as a substrate. And when it does, you don't get the Calvin cycle, you get what's called photorespiration. Because you haven't added any carbons, you can't generate any glucose. And so instead, you get this kind of inefficient side reaction where ammonia and carbon dioxide are produced, but you don't get any new sugar. That's a problem. It's a complete waste for plants to be using their rubisco in a way that doesn't generate sugar. C4 plants get around this by separating the influx of gases from rubisco in separate compartments. So C4 plants are a type of plant where the stoma, which allows carbon dioxide and other gases to enter, are separate from the cells that are actually going to run the Calvin cycle. And so you see there's 2 cell types here. There's the kind of light green cell, which is the mesophyll cell, and the dark green cell, which is the bundle sheath cell. And those are zoomed in over here. So in this picture, you see that carbon dioxide enters the mesophyll from the stroma nearby, and it comes into the chloroplast where it's converted into this 4-carbon malate molecule. Okay, so they're called C4 plants because they go through this 4-carbon intermediate, which can then be pumped using ATP into these lower bundle sheath cells where it's stored until it can be used and then used to regenerate carbon dioxide, which goes into the Calvin cycle to generate glucose for the cells. So in this way, carbon dioxide is entering in the mesophyll cell, and presumably oxygen is coming in too. But because carbon dioxide is being converted to malate and then moved into this other cell, the oxygen, which is found in the mesophyll cell, is separated from rubisco, which is found in the bundle sheath cell. And by separating oxygen from rubisco, you avoid the side reaction that otherwise would make rubisco really inefficient. CAM plants separate the influx of gases from rubisco by closing the stomata at times of high oxygen. So you tend to see this in plants like cactus, where the stomata are open at night. And so at night, carbon dioxide can enter. Oxygen will also enter, but it's not a great time of day for oxygen. The oxygen concentrations are lower at night. So carbon dioxide comes in at night. It's transferred from the chloroplast to the vacuole, where again, we generate this intermediate storage molecule. Here it's malic acid. And then during the day, the stomata are closed. There's no additional gas exchange happening here. And those malic acids from the vacuole can be transported back to the chloroplast, regenerate carbon dioxide, and then run the Calvin cycle, separating rubisco, which is present during the day, from oxygen, which, if it's coming in at all, is coming in when the oxygen concentrations in the environment are already lower at night. So C4 plants separate rubisco and oxygen spatially in 2 different cell types, and CAM plants separate oxygen from rubisco temporally at different times of day. And those adaptations allow them to grow even in places where the oxygen to carbon dioxide ratio is high.